Here I have a public dataset where human cells were infected with fluorescent wild-type or mutant Salmonella, and I am going to show you a colocalization analysis workflow that allows us to determine whether the bacteria have successfully reached the ER membrane and whether this changes when the bacteria carry a mutation. So I uploaded their images to our platform, and you can see here that we have 3 channels, one for the nuclei, one for the ER, and one for Salmonella. So the first thing we need is a cytoplasmic mask to restrict the analysis to the cellular region. In order to obtain that, I first segmented the nuclei using segment cell nuclei. I separated touching nuclei using watershed. Then I segmented the signal from the ER channel, and I used the nuclei as seeds to create a mask for each cell. I also added a dilation step to make sure to capture enough cytoplasm. Then I applied our Pearson Correlation node to calculate the correlation coefficients between Salmonella and the ER within these masks. Here you can see the results. Down here you can see an overview map of all the cells that were detected. Here I plotted the area versus the perimeter of the cell so that we can use this dot plot to create a gate to select a population of interest if we need to. In this table and in this bar chart you can see the Pearson correlation coefficient for ER versus Salmonella. You can see that this coefficient is higher when Salmonella is wild type versus when it is mutated, meaning that the correlation between Salmonella and the ER is higher for wild type cells.